All right, guys, give me just a second. Let me get attendance and all put in. All right, guys. Let me start out. I've already pulled this up, but let me pull it down real fast. And go into. All right. I just wanted to give you a heads up where I posted everything for the biotechnology project. This link here is going to show you some ideas of some different topics that you can choose from. You don't have to use this list. It's just there to help you if you can't come up with one on your own. So I want to click on that and just give you an idea. Again, there's tons and tons of topics on here for you to go through. So again, if you can't figure out something on your own, go and read through these and you should be able to get some kind of idea of something to do. It'll also help you reading through this to see really what biotechnology is, right? Again, it's using a microbe, make sure there's a microbe involved to do something for humans, whether that be in agriculture, in medicine, waste disposal. There's all kinds of ways that we use microbes to do things, right? Even our food that we eat. So again, I meant to say this the other day, but you need to approve your topic with me before you start. I don't want people repeating the same thing over and over again. So I will try and post a Google Doc and I'll put a link to it here in this area. So it's not here now, but I'll put a Google Doc where you can go in and type in your name, the name of your teammates, whoever's working with you, and then what your topic is going to be. That way I can approve it and I know we're all doing something different. It's not as important for this one as it is for the last one that we're going to do, because you're going to be presenting that one. And we don't want to sit and watch 10 videos over the same thing. It gets kind of boring. So again, I'll set up that Google Doc. Again, you got a while till this is due, but you need to start thinking about it, getting your team together, figuring out who you're going to work with. I'm not assigning them. You can pick whoever you want. If you don't know anybody, just put in here in the chat box. Just say, hey, is there anybody that needs a partner? And find somebody that way. I know somebody had emailed me the other day about needing a partner, so you might be able to get with them if you want to. So if there's anyone without a partner, just chime in on the chat box and let them know, and maybe exchange emails or phone numbers or whatnot, and go to it that way. All right, so there you go. You got several of you that need partners, so pick someone, get to know them. Again, preferably if you have lab with somebody that's in the class, that would probably be your best bet, but a lot of you don't. So kind of get together that way, share emails, and then you can maybe share your phone number or whatnot in private. There is a private chat on here feature. So if you want to do the direct chat to just one person, so you're not putting all your information out there, that would be perfectly fine. All right. There's somebody needing a third person. So use the direct chat and just talk to each other. Once you figure out, somebody that needs a partner, just direct chat them and exchange information and kind of go from there. All right. The project itself is here. What is biotechnology? This is basically just going to give you an overview of what it is. All right. So this will give you an idea of kind of what biotechnology actually is, so that you know what kind of topic to look for. Okay? So again, these documents are on here for you. And then the Dropbox is down here. Again, this isn't due till the 16th of March, so you still have quite a ways to go, a couple of weeks. It will sneak up on you though. I will warn you. All right. All right. So that's where you found that under chapter 9. We're going to finish up chapter 5 today, and I'm going to kind of go through some of these handouts that are here. These are the ones you can use on your test. So what we're going to be talking about first is glycolysis, respiration, and fermentation. And all of the information you need is in this document. Again, you can use this on your test. It's going to tell you how many ATPs are produced. I'm not wanting you to memorize all of these pathways. That's just a little bit above what we need for this class. If this was a biochemistry class, you would have to memorize all of them. I had to do it, but for this class, it's not necessary. So I'm giving you all of those. So make sure you download all of these and have them ready for your test when that day comes around. All right, so that takes care of business there. So let me pull up the slideshow and kind of finish out. Now, all right, so what we're going to start out with is carbohydrate catabolism. And this is better known as cell respiration or cellular respiration. Now, what I want you to gain from this is learning how many ATP are going to be made with this. Yes, you can work in pairs up to 3, just no more than 3. So if you want to work with a group of 3, that's fine, or a group of 2, that's fine. Just don't make it one person because it gets to where we have to watch too many videos and that type of thing. All right, so when we look at catabolism, or the breakdown of carbohydrates to release energy, typically we talk about glucose being the carbohydrate. That's kind of the crude oil of all living things. They have to convert it to ATP in order to use it like we do oil to gasoline. All right. So ATP is our gasoline, our cell's gasoline. And the same thing applies to bacteria. They use ATP just like we do. They're living things. All living things require ATP. All living things use ATP for energy. So it's a very important molecule. And we go through several steps in order to get the energy out of glucose. So if we go through glycolysis, we get a net of 2 ATP. And the reason I say net is because we get a gross of 4 ATP, but it takes 2 ATP to get it started. So we lose 2 ATP and we get 4 out. So we get a net of 2, right? So this is what I want you to know for your test. How many ATP are produced in each cycle? And again, you're not going to have to know all the biochemistry stuff involved with it, but I do want you to understand where things are made and why organisms typically want to use oxygen. All right. We're going to see in a couple of videos here in a minute, he's going to go through fermentation and then he's going to go through cellular respiration with oxygen. All right. If we don't have oxygen, we basically only go through glycolysis. All right. So we're only going to get 2 ATP net, and it's kind of a backup plan. So fermentation is not the way that most organisms want to get their energy because it's just a low yield. And what I mean by that is for every molecule of glucose, we only get 2 ATP. That's not very many. So it's kind of one of those things, well, if oxygen's not around and I have to do this, I'm going to go through fermentation in order to get enough ATP to survive. And that's really what they're trying to do. Now, some organisms are truly anaerobes, and they are only going to go through fermentation, right? Because they can't survive in the presence of oxygen. They never evolved the enzymes and things to make oxygen non-toxic. We don't think of oxygen being toxic because we breathe it in every day, but our body is able to take that oxygen and convert it into forms that are not toxic through our cellular respiration. So we have developed a way to take oxygen from the atmosphere and make it non-toxic and use it to gain energy. So we evolved that over time, but the first organisms were anaerobes. There was no oxygen. Think about it, before plants existed and photosynthetic organisms existed, there was no oxygen on the planet. It wasn't until they evolved that they started pumping the atmosphere full of oxygen, that all the other organisms had to adapt to that. And again, some did and some didn't. The true anaerobes never did evolve a way to live in oxygen. They just kind of hid out where there was no oxygen. But the other organisms evolved to use it, and eventually that led to multicellular organisms that we see today. So again, glycolysis, that is the only way that a truly anaerobic organism can get energy, and it's only going to get you to ATP. Now, if we have oxygen available, we're going to go from glycolysis into the Krebs cycle. And in the Krebs cycle, we're going to produce a lot of electron carriers. We're still not going to make much ATP, only 2 more, but we're going to create a bunch of electron carriers that are going to enter this electron transport chain. That's where we get the big payoff, right? So there we get 34 ATP. So if we look at the totals, we get 2 from glycolysis, 2 from the Krebs cycle, and 34 from the electron transport chain. Now, these numbers are theoretical, which means most organisms don't actually get this much ATP. They only maybe get a partial amount of this. So even in the video, when the guy's talking, he may use some different numbers than these. These are, in a perfect world, a microorganism could get 38 ATP. And eukaryotic organisms like us, we only get 36. We have a membrane-bound organelle, the mitochondria, and we have to shuttle some electrons across the membrane, and that requires 2 extra ATP. So we lose 2 of those, and we only get 36. But remember, in the bacteria, they don't have membrane-bound organelles. They are the organelle, and they are the one making the ATP, so they don't have to waste those extra 2. So they get a total, theoretically, again, of 38. All right, so we're going to go through anaerobic respiration first, and he's going to describe that in fermentation. And then we're going to watch another video where he goes through aerobic respiration. I like to use the videos because they have animation and you can see all the pathways moving and things going in and out. And I think that helps to comprehend what's really going on. But again, you don't need to memorize any of this stuff. You just need to know how many ATP are produced. And again, those are going to be listed on those documents that I provided for you. You just need to know how to read them and what pathway is what, right? So let's watch this, and then we'll come back and I'll talk a little bit more about anaerobic respiration. Then we'll look at aerobic respiration.
Hi everyone, Fritz here. In today's video, we're going to go over your anaerobic respiration. When we look at bacteria in a nutshell, the main objective of the bacteria is to produce energy. And whether this bacteria is an anaerobe or an aerobe, the primary process in which the initial ATP is made is always in an anaerobic fashion. And then after the initial ATP is produced, the bacteria, based off of its metabolic behaviors, will either continue down the anaerobic pathway or go down an aerobic pathway. And looking at those steps, the bacteria under normal circumstances will start off with a carbon source such as glucose. It will then put glucose through a process called glycolysis. At the end of the glycolysis process, we end up with 2 ATP molecules as well as 2 NADH molecules. Glycolysis will then convert the glucose to 2 pyruvate molecules, which are 3 carbon molecules each. And all of this is in a chemistry class. I just wanted to illustrate the fact that we are going to maintain the same amount of carbons going in as we do coming out. Now, if this bacteria is an aerobe, it will take the 2 pyruvate as well as the 2 NADH and put it through aerobic respiration, such as the electron transport chain, thereby creating anywhere from 32 to 36 ATP. However, we're focused on the anaerobic process. So what happens if the bacteria, which what we have here in this case, is an anaerobe? Well, in that case, the bacteria will go down the anaerobic pathway. And at the end of this process, the bacteria will either make alcohol or lactic acid. Either way, this process does not actually make energy, at least not enough energy worthwhile for us to count. But what's more important is that in this process of fermentation, the bacteria will actually take the 2 NADH from up here, convert it to NAD+, and then recycle it back into glycolysis. And the reason why this is so important is because these bacteria want to make the most use out of the resources that are available to it. So it doesn't want to waste anything, as was evident when we maintained our carbons from 6 up here to 6 down here. So this bacteria, if it wants to make 32 to 36 ATP as an aerobe will, it will actually have to repeat this process anywhere from 15 to 17 more times. And the most efficient way to do this is to recycle the NADH that it's not using and put it through glycolysis again, because it is a reactant in the glycolysis process when the bacteria finally makes the 2 ATP. But of course, for any one cycle of aerobic or anaerobic respiration, the aerobic will always create more ATP. But because this isn't available to the anaerobe, it has to go through this process. And here's some additional questions that you can have on test day. Now feel free to enter those questions down in the comment section below.
All right, so that takes you through anaerobic respiration. Now, again, notice on here, we're only making 2 ATP at a time, and we're recycling this NAD back into glycolysis. And it's just a cycle that goes through over and over again. But we're not getting a lot of energy, all right? Again, 2 ATP is not very much from a whole molecule of glucose. But what we end up with down here is alcohol or lactic acid. Now, if I were to take alcohol, which is the same stuff as in beer and wine, and I took pure alcohol and I light a match, it burns, and it burns very effectively. In fact, we use ethanol in our car. If you look at your gas, it's usually about 10% ethanol now. So it's still got a lot of energy trapped in this molecule, this alcohol that comes out of it. So we're wasting energy. So getting 2 ATP, making a high energy compound like alcohol or lactic acid isn't a very efficient process. But if there is no oxygen and you have to go anaerobic, you're going to do this. We do this as humans. When you exercise and you don't get enough oxygen to your muscles for it to go through aerobic respiration, it goes through fermentation and we produce lactic acid. That's what makes your muscles sore, right? So we do this as well when we have to. We don't want to, but if there's no oxygen or not enough oxygen, then we have to go anaerobic and start making energy that way, right? So this is one pathway, fermentation. Again, 2 ATP, very wasteful, not efficient, and really organisms don't want to do this unless they have to. Now, if I'm a true anaerobe, though, that's the only way I can do things, all right? I have no other mechanism. So you're going to have to go through this and get this little amount of energy out of whatever you're consuming. But that's your lifestyle. You're not going to be able to live in the presence of oxygen because it's toxic to you. All right, the next video is going to go through the aerobic side, and you're going to see we're going to make a ton more ATP than we do with the fermentation.
Hi everyone, Fritz here. In today's video, we're going to go over your aerobic respiration. Now, the last time we talked about respiration, we talked about it in the anaerobic aspect, where we have a bacteria where its desire is to get or make some kind of ATP. And we talked about this in the process of fermentation. But we also mentioned that in the process of making these 2 ATP from fermentation, we're also going to get 2 NADH. And these 2 NADH will help cycle back to the bacteria to repeat the fermentation process. And what we didn't talk about is bacteria that have the ability to use air to generate more than 2 ATP and 2 NADH. Now, whether the bacteria is an anaerobe or aerobe, it will always start off with this process. But if it is able to use air and has the presence of air, it will go down a cascade of events that will allow it to generate more ATP, specifically 32 ATP. So, how does it get here? Well, from here, we already know that our bacteria started out with a glucose molecule, which is a 6-carbon molecule. It will then break it down through a process called glycolysis. As we've already mentioned before, with anaerobic respiration, we start off with a glucose molecule. This glucose molecule will go through a process called glycolysis. And once we're done with glycolysis, our glucose will be processed into 2 pyruvate molecules. But in that process of glycolysis, we get our 2 ATP and 2 NADH. Now, what happens after this step can only be achieved by bacteria that are aerobes. Although the next step that we're going to go over, pyruvate decarboxylation, is technically still an anaerobic step. We take these 2 pyruvate and put it through pyruvate decarboxylation, which will convert our 2 pyruvate molecules to 2 acetyl-CoAs. And just like glycolysis, pyruvate decarboxylation gains us some energy here in the form of 2 more NADH. Now lastly, we're going to take our 2 acetyl-CoA molecules and put it through the last process of the electron transport chain, Krebs, or TCA, all of them meaning the same thing. And once our 2 acetyl-CoAs have fully gone through the electron transport chain, we can expect to get 2 more ATP, 2 FADH2, and 6 NADH. And now once we add all these things up together, you can see that we have 4 ATP, 2 FADH2, and 10 NADH.
Right. Now, in one of the diagrams that I let you use on your test, you're going to see it outline what each one of these is going to do. These are electron carriers. And what that means is each one of these has an electron that it can use to generate ATP in the electron transport chain. So even though we're only making a few ATP, 2 up here and 2 down here to make a total of 4, the important part is these guys. These guys are going to go in the electron transport chain, and that's where we're going to get the big bang for the buck. And we're going to get more and more ATP. But it's only because these guys can carry electrons into that system, right?
But you might be saying to yourself, Fritz, I thought we were going to get 32 ATP, not 4 in these other molecules that you mentioned, and I'm not sure about what the significance is. Well, their significance comes into play because we actually don't put the 2 acetyl-CoA through the electron transport chain. They do act as reactants, but what we're also doing in the electron transport chain is transporting the electrons from FADH2 and NADH. And once those electrons have been transported, we can expect that 1.5 ATP approximately and 2.5 ATP will be generated for each molecule. So this comes out to 3 ATP, and this comes out to 25 ATP. So no need to whip out your calculators, it's just simple addition. 4 plus 5, 29, plus 3 is 32. And that's how we get 32 ATP. Now, depending on the textbook and the actual processes that the bacteria go through, this number can vary, but you can expect it to be anywhere in the realm of 28 to 36. Because this is a microbiology class and not a biochemistry class, I really think it would be extremely cruel for your teachers to give you something like, how many ATP is created at the end of the electron transport chain? Is it 28, 30, 32, or 36? That to me would be extremely rare. In my experience, they'll give you some numbers that might have some correlation or no correlation at all, but they're going to be very off from this range right here. So they could say 2, which would be related to fermentation. They could also say 13, not related at all. They might even say 36, this would be the right answer, and then another wrong answer would be 22. 22 is not close to 28. So just remember 28 to 36, that's the range that you can expect when going through the electron transport chain.
All right, now for us in your textbook, just go with the number 38. All right, and again, that's going to be included in those documents that I posted. In fact, let me pull one of those up and show you what I'm talking about with that. Right, so this is going to give you an overview of carbohydrate metabolism or catabolism, and you're going to see the numbers on here. So glycolysis is going to produce 2. Let me skip on down to where I have it listed out. Here you have 2 ATP, right, being produced. There's the other 2. And then down here in the electron transport chain, I know I have it in here somewhere where I have all the numbers on there. Sorry, I'm skipping through so fast, but here. So again, here's our glycolysis. We produce 2 ATP. Our Krebs cycle, we produce 2 more ATP. And then through the electron transport chain, we produce 34. 2, 34 gives us a total of 38. So again, you have a cheat sheet right in front of you. So if I ask you any question like how many ATP are made during glycolysis, you can just look right here. It's 2. How many are made in the Krebs cycle? 2. How many are made in the electron transport chain? 34. How many are total? 38. You've got a cheat sheet right in front of you, right? So there's no need to memorize all this stuff. You'll have this available to you, all right? Just look through these documents, though, and make sure you know how to read them and where things are at so that you can easily find them when you're taking the test, all right? Pull this back up again. All right, so same thing I just showed you on that document is right here. So 2, 34 gives us a total of 38. Again, if we go over here through fermentation, we're only getting these 2, right? We're not going down through this because we don't have oxygen available, right? So no oxygen, we go through fermentation. With oxygen, we go through the Krebs cycle, the electron transport chain, and then we get that final total of 38. But again, if we do fermentation, we just simply produce 2 and recycle that NADH and continue that over and over again, trying to get a little bit of energy from that. All right, so I'm going to skip all of this because you really don't need to memorize all this stuff. You've got it on those documents. So again, just read through those and make sure that you know those numbers. So I'm not going to ask you like any of these intermediates that you see here. Again, if it's a biochemistry class, you'd have to memorize all this stuff, but it's not. So again, I've given you that cheat sheet, so it shouldn't be that big a deal for you to be able to figure out how many ATP are made where. I do want to go through chemiosmosis, though, just to give you an idea of how the ATP is actually made. So what we're going to do is pass electrons down that electron transport chain, and we're going to pump out hydrogen ions. So using the energy from the electron, we're going to pump hydrogen ions, and we're going to get a really high hydrogen concentration. Remember back from A&P, we talked about things moving from high concentration to low concentration. If I have a bunch of hydrogen up here and very little down here, they're going to want to move. And as they move, they go through this ATP synthase, which is an enzyme that converts ADP to ATP. So using the energy from this hydrogen ion falling back down through this, it literally spins almost like a turbine in a dam. As these hydrogen ions flow back down through this enzyme will spin and produce ATP from ADP. So this is the final end of the electron transport chain, right? So again, all along that transport chain, we're pumping out hydrogen ions, and then they fall back down there due to concentration gradient, and they move through the membrane, and they move through this ATP synthase enzyme, and that creates ATP from the ADP. So again, you can see the electron moving down the electron transport chain. Again, it just moves from compound to compound until finally we pump enough hydrogen out that it comes down. And notice this rotating motion you see here. Again, this enzyme just kind of spins around and produces ATP as it moves through. Just think of like a water going through a dam and turning a turbine to generate electricity, which is how we do that at our major lakes. It's like me, I live near West Point. You go down to the dam at West Point, when they generate power, they release water through the dam. And that water moving through spins a turbine, and that generates electricity. Same thing here. These hydrogen ions move through, and they cause this enzyme to be able to take ADP and produce ATP from it. Okay, so again, this is called chemiosmosis, where we take hydrogen ions and move them through and generate ATP from it. That's just another diagram showing you the ATP being made. So total of 38, but you have that cheat sheet. All you have to do is look on there. You'll be able to figure out all this stuff. All right. Now, fermentation, again, we talked about this, small amounts of ATP, but the final product still contains a lot of energy. So again, the ethanol or the lactic acid that we produce through fermentation still has most of the energy from that original glucose molecule. It's just stored now in a different form in ethanol or in lactic acid. Again, this is the backup plan that organisms want to go through when they don't have oxygen available. So it doesn't use the Krebs cycle, it doesn't use the electron transport chain. And basically it's just kind of trudging along, hoping it can make enough energy to survive until oxygen becomes available again. Or if I'm a true anaerobe, that's all I can do. Can't do anything else. All right, so fermentation produces, again, lactic acid. And I want to show you this slide because this is how yogurt is made. You may not realize how easy it is to make some of these foods that we eat, but yogurt's extremely easy to make. All you have to do is take milk, and you need to warm it up, add a little bit of yogurt that has live culture in it, and overnight it will turn into yogurt. Believe it or not, that's all it takes. All right. These lactobacillus bacteria are going to take the sugar in the milk, and it's going to convert it into lactic acid. And that's what we taste when we eat yogurt. We get that tart taste, that sour taste that we get. The longer you let your yogurt ferment, the more lactic acid is made and the tartar it gets. The store-bought yogurt that you buy, make sure when you buy it, it says live cultures on it, because those are good bacteria. You want those. You don't want to sterilize. If they pasteurize the yogurt, it's not very good for you, and you don't get the benefits from eating it. So you want those live cultures. And if you want to make yogurt, just buy a little tub at the store, take some milk. Again, you're going to heat it up and keep it at a temperature. And then that bacteria is going to consume the sugar and convert it to lactic acid overnight. It'll thicken, and then you'll get whey, which is kind of a liquid that forms on top of it. You drain that off, and then the rest will be yogurt. And it's so inexpensive and so good for you, and you can flavor it any way you like. Once you learn to make it, you'll never buy store yogurt again. Make it all the time, and it's easy. They even have it, I guess Walmart or even Amazon. You can buy a yogurt maker that will keep it at the right temperature, and all you do is pour milk in it and add live culture, and you're off to the races, and it'll make it for you. It's really simple. So again, this is fermentation, and these are the bacteria that ferment and create lactic acid. Now, alcoholic fermentation is going to produce ethanol and CO2. And glucose then is oxidized to pyruvic acid, and then that's converted to acetaldehyde and CO2. And then acetaldehyde is reduced to ethanol. Now, what happens with this, and the reason I want you to be aware of this acetaldehyde is when we metabolize alcohol, let's say we drink it, we basically reverse this reaction and we take alcohol and we convert it to acetaldehyde. Alcohol itself is not necessarily toxic to the body, but acetaldehyde is. And so when we break down alcohol in the liver, we produce acetaldehyde, and that's what gives the effect of having a hangover. That's what does the damage to your liver. That's what does the damage to your body. It's not the alcohol, but it's the stuff that is converted to when we metabolize it. So basically, we have alcohol dehydrogenase, which is an enzyme that converts alcohol back to acetaldehyde. And again, that's the dangerous part. So again, there's certain products on the market now that they are advertising that's supposed to bind with acetaldehyde and convert it into something less toxic. I don't know if you probably need to believe that or not. I don't know. It's probably a gimmick. But at least the science behind it makes sense that you're trying to get rid of this compound because that's the one that's dangerous. But again, the yeast and other microorganisms can take sugar, they can convert it into alcohol, and again, alcohol is still a very high energy molecule. Right, so through fermentation, again, we can produce lactic acid or alcohol, but the whole goal of this is to get this NADH and put it back into the system. So we're basically just recycling that so we can keep making this small amount of ATP. Now, alcohol and lactic acid aren't the only products of fermentation. There's specialized bacteria that can do other things. So if we look at, Streptococcus, Lactobacillus, Bacillus, the ones we use to make yogurt, they make lactic acid. Saccharomyces, which is yeast, it makes ethanol. But we have other bacteria that can make other things for us that we can use in, let's say, industrial applications, right? So propionic acid, acetic acid, which is basically vinegar, butyric acid, butanol, acetone, isopropyl alcohol, all of these things we use in the industry, and we even use ourselves. A lot of you still use acetone for fingernail polish remover. You shouldn't do that because acetone is a carcinogen and it goes right into your bloodstream. So you need to stay away from that if you can. Isopropyl alcohol, that's your rubbing alcohol. Same stuff you use at home all the time. Over here, we see ethanol, lactic acid, succinic acid, acetic acid, formic acid, butanediol, acetoin. So again, alcohol and lactic acid aren't the only two products of fermentation. It's just they're the most common. Right, so you can see some of the uses for all of these products. So again, acetone, butanol, pharmaceuticals, methane, we use that as fuel. Sorbose, we convert that to ascorbic acid, which is vitamin C. So we use all these microorganisms to make these products. Now, if you want to, you can use this chart, and this is biotechnology at its best. We're using a microorganism to make something that we need, all right? So again, we make pharmaceuticals using Clostridium. We make methane fuel. And again, we make all of these products that we use. So that is a form of biotechnology. Right. Now, all of this I'm going to skip over because you have a diagram that shows you this. But this is just showing you how lipids and proteins are broken down. The goal of all of this is to get to the Krebs cycle. All right. So if I eat a lipid or a fat, or a bacteria consumes a lipid or a fat, it's going to break that down. And the goal is to get this acetyl CoA. And you're going to see this with every type of biological compound that we consume or bacteria consume. So here's lipids, right? And here you can see proteins, carbohydrates, lipids, they all go to the same place. They all want to go into the Krebs cycle so they can go into the electron transport chain and we can get energy from that. So it's not just glucose that we get energy from. We get energy from all kinds of sources. So again, bacteria, humans, dogs, cats, every living organism does these same things. They take these biological compounds that they consume and they convert it over into acetyl-CoA and enter the Krebs cycle and then make ATP from that. Right, now in lab, you're going to start doing some biochemical tests after your midterm. So once you get in the second half of the course, you're going to start doing some different stuff than you've been doing in lab. So you're going to do all kinds of different tests, and you'll use indicators and things to give you a color change to show that something has happened. So you can identify bacteria by detecting what enzyme they have. We don't do it like we used to with the Entera tubes, but they were basically these tubes. I went over them before that have all these different reactions that you can look at, and it gives you a number, and you can identify the bacteria based on what they can consume. And what the bacteria you can consume is based on what enzymes they produce. So fermentation test, you can look at who ferments what. Can it ferment this? Can it ferment that? With your dichotomous keys, remember you had some of that involved with it. It says, can it ferment, citrate? Can it ferment this? So again, we can do all these biochemical tests and see if it has this enzyme, that enzyme, and it helps us identify the bacteria. We can look for other enzymes like oxidase and identify the bacteria that have cytochrome oxidase, so Pseudomonas, that would help identify it. Right. So kind of switching gears, we're going to go to photosynthesis. I'm not going to get too deep in the woods with this one either. And you have a diagram. Let me go back with photosynthesis on it. So you can go here and click on that. Again, you'll have a cheat sheet and it'll talk about cyclic and non-cyclic, which we mentioned before. And then it goes through and shows you all the different pathways. So again, you don't need to memorize this. You have a cheat sheet in front of you, and you should be able to look on here and answer any question that I ask you about. Just make sure you read through these ahead of time so you know where to look for this stuff. Again, photosynthesis, cellular respiration are pretty much the opposites of one another, but they're both used to get energy, and that's the thing. Things that photosynthesize use the energy from the sun to produce carbon-containing compounds that they can use to make ATP, and then heterotrophs like bacteria that eat other things, they are going to eat those products produced by the plant or produced by an animal that ate a plant. But all this is just taking that energy from the sun and trying to get energy for the organism out of it. All right, so there's two types of reactions that we see in photosynthesis. We have the light-dependent reaction, and that's where we convert light energy into chemical energy, right? And then we have the light-independent, sometimes called the dark reactions. ATP and NADPH are used to reduce CO2 to sugar, and we use that to make carbon compounds, right? So we look at photosynthesis, the light reactions are going to occur, of course, in the daytime. The dark reactions are going to occur at night. Photosynthesis is kind of unique in that these organisms take the energy from the sun and they make compounds, and then they use those compounds later to generate ATP. So they are taking an extra step that we don't have to do. So just like we are heterotrophs, bacteria that are heterotrophs, they eat other things. They don't have to go through the capturing of sunlight and converting it into sugars. We just simply go out and eat those sugars produced by something else. So we eat a plant, or we eat an animal that ate a plant, and then we get the energy that way. So we skip this whole step of taking light and converting it over into carbon-containing compound. So there's two types of photosynthesis, and this you'll be asked on your test, but oxygenic, right, is going to produce oxygen. That's the normal photosynthesis we all think of. Anoxygenic is going to be one that uses something else other than oxygen to capture electrons, right? So we're still taking CO2, and that's the carbon compound that we're going to use to make sugars. But instead of water, in this case, we're going to use hydrogen sulfide and light energy, and we're going to create glucose, water, and sulfur. Up here, we're taking CO2 and water, which we're used to, like plants do, and making glucose, water, and oxygen instead. So this is where we get the oxygenic. We produce oxygen and oxygenic. We use some other method. Now, this one is a lot more rare than this one. This is the norm, the oxygenic. This is what we think of with all the plants that live around us. And you were probably taught in grade school and high school about photosynthesis. You take CO2 and you make oxygen and sugar from it. Down here, these are some unusual bacteria that can take hydrogen sulfide, which is that rotten egg smell, and convert that into sugar and then sulfur instead of oxygen. All right, so we went over this before, but cyclic photophosphorylation, we take light, we boost an electron, it moves down the electron transport chain. We use that energy to make ATP. That electron goes right back to chlorophyll, gets boosted by light again, and that continuously happens over and over again. Non-cyclic, we have to get the electron from somewhere else. And in this case, we get it from water, right? So we're going to extract 2 electrons from water, put that into chlorophyll. That's going to get boosted, go down the electron transport chain, make ATP. Then it goes into chlorophyll again, and we excite them one more time with light. And then that's going to come down and give off the energy, and then that electron is going to now be lost, and it's going to be lost to NADPH. All right. So we have to come back and replenish this all the time with water. And that's why plants require water. That's why any photosynthetic organism requires water. They have to have it in order to be able to recharge the system, because they need those electrons to go back in and start to go through the cycle again. Right? So again, we go boost it up, make ATP, we excite it again in the presence of light, and that electron is going to be lost to this NADPH, and we have to replenish it with water to go through the cycle again. Right, so in this step, we call it a Calvin-Benson cycle. This is where we actually take the ATP that's made in photosynthesis, and we convert that into glucose and other sugars. So this is where the energy is going to eventually be stored, is in these glucose molecules. Now, for a heterotroph like us, we are going to eat the plant and get this glucose and other sugars from them. All right. So we allow the plant to go through all of these chemical reactions and make these sugars, and then we take the energy in that sugar and we use that for our nourishment, right? And bacteria that are heterotrophs do the same thing. They basically eat organic material, whether it be glucose or lactose or something else, that's been made by a photosynthetic organism, or at least initially was made by a photosynthetic organism. All right. Now, this is one that you definitely need to memorize, right? So pay attention to this, and I will go through all of this with you. All organisms fall into one of 2 groups. Are they chemotrophs or are they phototrophs? Which means, are they getting chemical energy like we do from eating something else, or do they use light to create organic compounds, right? So if we look over here, let's just take the left side first. The chemotrophs consume energy, all right? And that carbon source is either organic compounds, like we do, we eat food, and we get our organic compounds from the food. And then depending on which way we go, we either use oxygen like we do, all animals, we're animals, fungi, protozoa, and bacteria, all are going to use oxygen here. If they don't use oxygen, again, they're going to either ferment or they can use a version of the electron transport chain, right? But what you notice over here is that we're consuming chemicals already made by a photosynthetic organism. We go on this side, we have photoheterotrophs, which take organic compounds and then they use light to help break the bonds in that and get energy from it. Or if they're photoautotrophs, they're going to make their own energy. All right. And again, it can be oxygenic photosynthesis or anoxygenic photosynthesis. Right. So they can go down one or two pathways. So Make sure you know this. I think I have also put a diagram similar to that here. Yes. So you can have this as your cheat sheet. Just make sure you know how to read this and know who uses what for energy and whether they use oxygen or not. Um, Again, do they use chemical energy or light energy? And do they use oxygen or not use oxygen? So again, you kind of have a cheat sheet here. It's 1 page long. Print that out and you'll have that available to you. Right, so we'll skip through that. There are some unusual bacteria. They're not medically significant, but I do want to just mention them. These are rare organisms that actually live at the bottom of the ocean, miles down. The water is superheated above the boiling point, and they're able to live here. And they use energy from inorganic chemicals, and they're able to basically become the base of a food chain without light. And we don't know of anything like this, or we didn't know of anything like this on Earth until we started exploring the deep sea. We thought everything that was alive on the planet got its energy from sunlight. We now know there's these rare organisms that can live around these deep sea volcanic vents. They can actually use the chemicals that are coming out of the ground for energy instead of having to use light energy. And we call these chemoautotrophs. So instead of using light like a photoautotroph, these chemoautotrophs can use these chemicals coming out of these volcanic vents excuse me, for energy. So just a side note, again, they're not medically significant. They don't infect people. So really not super important for this class, but just as a side note, there are these unusual guys that are out there. So this is going to go through chemosynthesis and just give you an idea of how these little guys live in such extreme environments.
Hi, this is George coming to you from Nautilus Mission Control at the University of Rhode Island's Inner Space Center. The second leg of the 2012 season brings Nautilus back to the Anaximander seamounts, where something surprising awaits deep down where the sun don't shine. Life. Now, at or near the surface of our planet, all life is powered by the sun in a process called photosynthesis. Plants capture light energy and store it in chemical bonds. Then animals eat those plants. So at the bottom of any food chain, you'll find photosynthetic organisms. But what about deep underwater where there is no light? How can life survive? The answer is chemosynthesis. Now, chemosynthesis isn't actually that different from photosynthesis, but instead of light energy powering life, chemical energy powers it. Methane and sulfides that bubble up through the Earth's crust are used by chemosynthetic bacteria. That releases energy which powers a process similar to photosynthesis. Organisms then either eat the bacteria or provide a home for them within their tissue and then use the energy that the bacteria produce. These two worms, for example, have no mouth and no gut. They don't eat like most animals do. Instead, they depend entirely on chemosynthetic bacteria to survive. So there are two parallel food chains on this planet. One that gets its energy from the sun, and another that gets its energy from molecules that seep out of the Earth's crust. And this second food chain was discovered only 35 years ago by a team that included our very own Dr. Robert Ballard. Well, we hope you enjoyed this peek behind the science. For more, be sure to follow us on Facebook and Twitter, and of course, you can always join the expedition live, right here at nautiluslive.org.
All right, so that just gives you a little insight into that second way that organisms can survive on this planet. Again, just 40 years ago, we had no knowledge of these things. We didn't know that there were any living organisms that could possibly survive without sunlight. So that gives us hope that maybe on other planets that there are organisms that can carry out chemosynthesis and they may not need sunlight and they can live inside of rocks and things like that that provide us some shelter from radiation and that type of thing. All right, this is a good chart to look at just to give you an idea of what a photoautotroph is, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. So these are the ways that organisms get energy. So a photoautotroph it's going to capture light, right? So these are going to be the ones that can photosynthesize. A photoheterotroph is also going to use light, but instead of CO2 as its carbon source, it's going to use other organic compounds, right? So much rarer. These are more prevalent. The chemoautotroph uses some kind of inorganic chemical like we just saw in the video in CO2, and they can get energy that way. And then a chemoheterotroph, they get chemical energy. So these are things that eat other organisms, just like us, right? The carbon source is the organic compounds they eat. So again, animals, protozoa, fungi, bacteria, fermented bacteria, all of these are going to use organic compounds created by one of these photoautotrophs up here, or a photoheterotroph, or a chemoautotroph. So all of these are bases of the food chain, and then these guys take advantage of it. Right? All right, so this stuff is not important. I'm going to skip over it. And you don't need to know any of that. And then the last
A
thing.
good breakfast begins with Bob's Red Mill from the passionate purist.
The last thing we're going to look at, and then we'll have a little short quiz, is just an overview of metabolism. All right, so this is just going to go over everything we've talked about and put it all together in one place.
In the second segment of bacterial metabolism, we'll look at the electron transport chain. You can use a yellow highlighter. The electron transport chain is made up of molecules of increasing electronegativity. When you hear me say increasing electronegativity, all you really need to think of is stronger and stronger magnet. So if the electrons are attracted to the magnets, and each magnet is stronger than the one before it, then the electrons are going to go hop, 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 down to the biggest magnet of all, which is either oxygen, or in humans, or maybe it's nitrogen or even sulfur. So down here, this is the strongest magnet. Strongest electron magnet. Loves to eat them. Gobble, 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 gobble. And we call it the final electron acceptor, because who's going to be the final one? Well, the biggest magnet. So the final electron acceptor, and it's either oxygen or nitrogen or sulfur or possibly some other strong electron acceptor, although these are the ones I'm most familiar with. So what happens is each magnet is bigger than the one before, so the electrons are pulled to the first one, and then the next magnet takes it, and then the next magnet takes it, and they start flowing down this chain. And what happens then is, as they're pulled to the final electron acceptor, that the movement, just like a hydroelectric dam, provides the energy to power up this machine called ATP synthase. ATP synthase starts churning out ATP as electrons flow by it. Lots and lots of ATP. Lots. So the flow of electrons moves the ATP maker machine, which is ATP synthase.
All right, that just gives you a final little overview of metabolism. So we don't need to see. All right, So I do have a quiz. So we have about 15 minutes left. So you should be able to easily get that done before the end of class. So let me show you where that's at and turn it on so you can get to it. So go where you normally go with the daily quizzes. And this one should be daily quiz 4. And I'm about to make it visible. So you should be able to get in it. It's due at 7. You have plenty of time. You got 30 minutes to get it done. So make sure you go through and answer all of those correctly. If anybody has any questions, I'll hang around for a few minutes and answer anything. Again, if you need to find a partner, just email or direct message back and forth and figure something out how you can communicate and get with each other. Again, you don't really have to meet in person to do this. You can do it all digitally. There's so much software out there now that allows you to communicate. You got Zoom, you got WebEx, you got all of that. You can do FaceTime on your phone. Just all kinds of ways to communicate. So if anybody has any questions, I'll stick around. If not, go ahead and knock out Daily Quiz 4, and then I'll see you back on Thursday.